Hello guys, welcome to my channel. Today I will discuss about the role of growth factors in cell cycle, DNA replication in S phase and the role of ATM, ATR and P53 in DNA damage checkpoints in detail. To know about cell cycle phases, define checkpoints and MPF or maturation promoting factor or about cell signaling in detail, please watch my previous videos. Links provided in the description. So let's start. Let's first understand how growth factors regulate cell cycle at G1 phase. We have discussed in my earlier videos that how proliferation of animal cells is regulated largely by extracellular growth factors which control the progression of cells through the restriction point in G1. In the absence of growth factors, cells cannot cross the restriction point and enter the quiescent or resting G0 stage. This suggests that though the availability of growth factors, the growth factors receptors turn on downstream signaling pathway leading to regulation of cell cycle at G1 phase. One connecting link is cyclin D. Cyclin D is synthesized in response to growth factor stimulation. This occurs by the signaling through RAS-RAP MEK-ERK pathway or the very known MAP kinase pathway. This cyclin D continues to be synthesized as long as growth factors are present. However, cyclin D is also rapidly degraded due to ubiquitination by the APC ubiquitin ligase. APC stands for anaphase promoting complex. So its intracellular concentration rapidly falls when the growth factors are removed. I have discussed MAP kinase signaling pathway in my signaling videos. This means as long as the growth factors are produced, cyclin D remains available to complex with CDK4 and 6, which allows crossing the restriction point in G1 and the cell cycle to continue. Without growth factors, there is no cyclin D and thus the cell cycle halts at restriction point and enters G0 phase. So if any defects occur in cyclin D resulting in losing its regulation by growth factors, then uncontrolled cell cycle will occur leading to cancer. Another important protein is the key substrate protein of cyclin D CDK4/6 complex is retinoblastoma or RB. Mutations in the RB gene leads to a rare inherited childhood eye tumor. We can see this in this picture. RB is a tumor suppressor gene. Inactivation of tumor suppressor gene leads to tumor development. These tumor suppressor genes act as brakes in cancer. The RB plays important role in cell cycle progression. In normal unphosphorylated state, the RB remains attached or bound with a transcription factor called E2F that regulate expression of many genes involved in cell cycle progression like cyclin E. The cyclin D CDK4/6 is responsible for the phosphorylation of RB after the cell cycle crosses the restriction point. When bound to E2F, the RB acts as a repressor of E2F and prevents functioning. Now, phosphorylation of RB by cyclin D CDK4/6 leads to the dissociation of the RB from the E2F. As a result, the E2F becomes free and activated and thus transcribes genes such as cyclin E. Thus, growth factor also regulate the entry into S phase by synthesizing cyclin E through the availability of cyclin D. In the G1 phase, the cyclin E CDK2 complex remains inactive due to the presence of a CDK inhibitor P27. Through the G1, the growth factor signaling pathway inhibit the synthesis of P27, which results in the activation of CDK2 at the end of the G1 phase. Also, the bound P27 is also degraded by the activation of CDK2. The cyclin A CDK2 now becomes hyperactive, which also inhibits the APC ubiquitin ligase that prevents degradation of cyclin E. Now let's understand in detail the DNA replication in S phase. S phase is initiated by cyclin E-CDK2 that activated DNA synthesis at replication origins. In mammalian cells, there are thousands of replication origins. It is essential that replication at each origins occur only once during each cycle, unless there will be catastrophic conditions where equal division of genome will not be possible. Suppose the 2n genome is replicated twice instead of once in a cell during S phase, then we will have 8n instead of 4n. As a result, after mitosis, we will have two cells having 4n instead of 2n genome each. Thus, it is important that only one round of replication occurs per cell cycle. Here comes the role of MCM helicase. During G1 phase, the MCM helicase together with the origin recognition complex or ORC binds to the origin of replications and form the pre-replication complex, which is inactive. The MCM helicase becomes activated by the action of cyclin E-CDK2 when the cell cycle enters the S phase. Now, activation of MCM initiates the DNA replication, after which the MCM dissociates. During S, G2 or the M phase, the MCM helicase cannot bind to the origin of replication. As a result, only once in G1 phase the MCM helicase binds to the origin of replication, so only one replication cycle occurs per cell cycle. Now finally, let's discuss DNA damage checkpoints in cell cycle arrest. I have discussed about cell cycle checkpoints and MPF in my previous video. DNA damage checkpoints play a critical role in maintaining the integrity of the genome. They arrest cell cycle progression in response to damaged or incompletely replicated DNA. These DNA damage checkpoints operate in G1S and the G2 phases of the cell cycle and allow time for the damage to be repaired before DNA replication or cell division proceeds. This is mediated by two protein kinases called ATR and ATM that are activated in response to DNA damage. ATR and ATM then activate a signaling pathway leading to cell cycle arrest and also activation of DNA repair and in some cases programmed cell death. ATR and ATM are components of protein complexes that recognize damaged or unreplicated DNA. ATR is activated by single-stranded break or unreplicated DNA, while ATM is activated by double-stranded breaks. Once activated, both ATR and ATM phosphorylate and thus activate checkpoint kinases CHK1 and CHK2 respectively. These CHK1 and CHK2 phosphorylate and inhibit the protein phosphatase CDC25. As CDC25 phosphatases are required for both CDK1 and CDK2 activation, their inactivation leads to cell cycle arrest at G1S and G2 phases. Another tumor suppressor protein P53 mediates cell cycle arrest at DNA damage checkpoints. We know that P53 is a tumor suppressor protein. That means its role is to suppress uncontrolled cell growth and thus prevent cancer. Activation of ATM and CHK2 due to DNA damage phosphorylates the P53. This phosphorylation stabilizes the P53, which in turn prevents degradation by MDM2. Stabilization by phosphorylation leads to rapid increase in the P53 level. This P53 is a transcription factor that transcribes a CDK inhibitor called P21. This P21 now binds to the cyclin E-CDK2 and thus arrests the cell cycle progression by inhibiting the complex. This way, P53 blocks the cell cycle until the damage is repaired. When the DNA damage is repaired, ATM and checkpoint 2 level drops, leading to degradation of P53 and thus P21. Without the P21, the cyclin E-CDK2 complex becomes activated and the cycle enters the S phase and continue. So, Today I have discussed about the role of growth factors in cell cycle, its role in E2RB E2F complex, DNA replication in S phase, and the role of ATM, ATR and P53 in DNA damage checkpoints in detail. To know about cell cycle phases, define checkpoints and MPF, or about cell signaling in detail, please watch my previous videos. Links provided in the description. In the meantime, don't forget to like, share my videos and also subscribe to my channel. Thank you. Stay home, stay safe and study online.